What you're watching now is Big Tech, and the underlying premise to this show is trying to illustrate what biologic internet gateway technology is. And what that is a fusion between human biology and the internet as we understand it. So if you go to this page, you'll see TMC down here and a little Twitter bird, and click that and get to Twitter. And the notes, there's an archive notes page over here, and it's on the upper right corner. If you're on a mobile device, you'll probably have to scroll down. It'll be center screen, but you'll scroll down if it's a phone or a tablet. And these notes are going to parallel or correlate to each episode. And this episode is 33. And I'll read this, and then we're going to get going. So, Big Tech. Smart devices, IoT, big data, artificial intelligence, and machine learning are with each passing day more and more interwoven within all aspects of our society and daily lives. The digital space is maturing into an environment capable of fully immersive, augmented, and virtual reality experiences. Unlocking and internetworking the human mind is no longer science fiction. Our species is transitioning through another giant leap in our evolution. The language of the nervous system and brain is being decoded and digitized, thus becoming capable of being transmitted over modern communication networks. Together, we will explore modern telecommunication architecture, neurotechnology, and novel medical applications that can replace conventional treatment options. Humanity will continue its epic struggle between the preservation of liberty and privacy versus global oppression and enslavement. We must decide whether we retain our free will or become truly domesticated through technologic mind control. So when you go to the link page here in PSB, you'll see a title here, Read, Watch, Think for Yourself. And if you click that Big Tech Notes archive, it'll take you to this page. And it's a link to all the video archives, or excuse me, all the note archives. So we're on episode 33 today, and this document that's a share file basically will take you through episode 31 through 40. And right now we're on episode 33. And with that, we will start.
Humans and machines have been merging for thousands of years. Right now I'm wearing shoes, I have a microphone on my jacket. We all probably used our phones at least once today. And we communicate with the augmentation of all sorts of amplification and even translation technologies. You can speak into a machine and it'll translate the words you're saying in nearly real time. So I think what might be different in the years to come is a matter of degree, not a matter of kind.
A matter of degree instead of a matter of kind. So what he's basically saying is that we are already dependent and or interconnected with machines. And just ask yourself, how long or how far can you go in your day without your cell phone? Or referencing something on a computer? Or it's even as advanced or as implemented or integrated as your vehicle. Your vehicle is full of computers.
One concept that I think is emerging is what I like to call the brain coprocessor, a device that intimately interacts with the brain. It can upload information to the brain and download information from it. Imagine that you could have a technology that could replace lost memories, or augment decision-making, or boost attention or cognition. To do that, though, we have to understand how the brain works at a very deep level. Although over 1/3 patients have had brain implants or neural implants that stimulate the nervous system, so far they've operated in an open loop fashion.
So we're going to go on a ride tonight, and the next few episodes are going to really, I think, stretch the realm of possible. The way that I'm going to attempt to frame this, but it really is the idea of hijacking or understanding biology to the extent with such specificity that integration into the internet is possible at this point. And what this gentleman is talking about, who is probably one of the world's best at what he does. He works at MIT. He's a professor and he's a researcher and designer. DARPA awardee, et cetera, et cetera. His name is Edward Boyden. And what he's talking about is literally having AI in your head all the time. as a co-processor, implying that your brain is already a processor.
That is, they drive activity in the brain, but not in a fully responsive fashion. What we want to do is to have bidirectional communication to the brain. Can you read and write information continuously and supply, maybe through coupling these interfaces to silicon computers, exactly the information the brain needs? My hope is that over the next 5 to 10 years, we're going to get deep insights, thanks to our technologies, into how brain circuits compute.
So this is a lot to unpack in one episode, right? We're going to do this in phases. We've been doing this in phases for those of you that have been tuning in over the last few months. There's a lot to go through. There's hardware and software, and then putting it all together, and then biology. All of those pieces need to be addressed. So we'll do it a piece at a time.
And that will drive the design of these interfaces so that we can deliver information to the brain and record information from the brain using the natural language of the brain, reading and writing information in a way that augments, for example, the number of things you can hold in your mind at once, or the ability to recall things nearly perfectly, which is, not an ability so different from looking something up on a search engine on your phone, right? So I think what's going to happen is a continuation of this trend. And I think a lot of people like to talk about artificial intelligence right now. Artificial intelligence, as it stands, is based on a lot of concepts that go back many decades.
All right, so cell phone, right? We kind of set the table, like that's where we're at now. Artificial intelligence, he's beginning to discuss where that is and where it needs to go. And this is where it, in my view, this is where it gets kind of, we're blurring this line between organic and what we would consider real and artificial, right? That line is becoming blurred.
That build from some very simple observations about the brain. What might AI do, though, once we have incredibly deep insights into the nature of creativity and ethics and other things that the human brain seems to be uniquely equipped for? I think once we start to couple artificial intelligence to the brain that can really augment these uniquely human capabilities, it leads to a new era of what you might call hybrid intelligence. So it won't just be AI running away in some positive feedback loop. It won't be humans upgrading themselves in the absence of coupling to the world, but I think it'll yield a new kind of symbiosis. And I think that's probably the best possible path.
Kind of sounds like Elon Musk, doesn't it? A symbiosis with AI. What is AI? And everybody has their own understanding of what AI is. And I think that is AI. Is it could be anything, but it's who builds it. That's what's going to ultimately define it.
And it's also already, if you look at how people operate in the world, what seems to be one of the most prevalent models.
Okay, we're going to move forward. So this is 2016, March 8th. Article published. The U.S. government launches a $100 million Apollo project of the brain. Intelligence project aims to reverse engineer the brain to find algorithms that allow computers to think more like humans. 3 decades ago, the U.S. government launched the Human Genome Project, a 13-year endeavor to sequence and map all the genes of the human species. Although initially met with skepticism and even opposition, the project has since transformed the field of genetics and is today considered one of the most successful scientific enterprises in history. Now, the Intelligence Advanced Research Projects Activity, IARPA, a research organization for the intelligence community modeled after the Defense Department's famed DARPA, has dedicated $100 million to a similarly ambitious project. The Machine Intelligence from Cortical Networks program, or MICRONS, aims to reverse engineer 1 cubic millimeter of the brain, study the way it makes computations, and use those findings to better inform algorithms in machine learning and artificial intelligence. IARPA has recruited 3 teams, led by David Cox, a biologist and computer scientist at Harvard University, Tai-Sing Lee, a computer scientist at Carnegie Mellon University, and Andreas Tolies, a neuroscientist at Baylor College of Medicine. Each team has proposed its own 5-year approach to the problem. And that 5 years is just about expired, folks. It's a substantial investment because we think it's a critical challenge, and it'll have a transformative impact for the intelligence community as well as the world more broadly, says Jacob Vogelstein at IARPA, who manages the Microns program. Microns, as a part of President Obama's BRAIN initiative, is an attempt to push forward the status quo in brain-inspired computing. A great deal of technology today already relies on a class of algorithms called artificial neural networks, which, as their name would suggest, are inspired by the architecture, or at least what we know about the architecture of the brain. Thanks to significant increases in computing power and the availability of vast amounts of data on the internet, Facebook can identify faces, Siri can recognize voices, cars can self-navigate, and computers can beat humans at games like chess. These algorithms, however, are still primitive, relying on highly simplified processes of analyzing information for patterns. Based on models dating back to the 1980s, neural networks tend to perform poorly in cluttered environments where the object the computer is trying to identify is hidden among a large number of objects, many of which are overlapping or ambiguous. These algorithms do not generalize well. Seeing one or two examples of a dog, for instance, does not teach the computer how to identify all dogs. Humans, on the other hand, seem to overcome these challenges effortlessly. We can make out a friend in a crowd, focus on a familiar voice in a noisy setting, and deduce patterns in sounds or an image based on just one or a handful of examples. We are constantly learning to generalize without the need for any instructions. And so the Microns researchers have turned to the brain to find what these models are missing. That's the smoking gun, Cox says. While neural networks retain elements of the architecture found in the brain, the computations they use are not copied directly from any algorithms that neurons use to process information. In other words, the way in which current algorithms represent, transform, and learn from data are engineering solutions determined largely by trial and error. They work, but scientists do not know why. Certainly not well enough to define a way to design a neural network. Whether this neural processing is similar to or different from corresponding operations in the brain remains unknown. So if we go one level deeper and take information from the brain at the computational level and not just the architectural level, we can enhance those algorithms and get them closer to brain-like performance. So they're going to copy how the brain functions and apply it to artificial intelligence to allow for a more robust generalization. Our ability to generalize and form opinions, basically, or educated guesses or assessments. That's what they're looking for artificial intelligence. The various teams will attempt to map the complete circuitry between all of the neurons of a cubic millimeter of a rodent's cortex. I will read that statement again. The various teams will attempt to map the complete circuitry between all the neurons of a cubic millimeter of a rodent's cortex. This volume, which constitutes less than 1/millionth the size of a human brain, may seem tiny, but to date, scientists have only been able to measure the activity of either a few neurons at a time or millions of neurons contained in the composite pictures obtained through functional magnetic resonance imaging. The members of Microns plan to record the activity and connectivity of 100,000 neurons while the rodent is engaged in visual perception and learning tasks, meaning it's alive. A relatively enormous feat, since it requires imaging with nanometer resolution, the twists and turns of wires whose full length is a few millimeters. That's like creating a roadmap of the US by measuring every inch, Vogelstein says. Still, Vogelstein is optimistic because of recent support given for large-scale neuroscience research. With the advent of the BRAIN initiative, an enormous number of tools have come online for interrogating the brain, both at resolution and scale that's required for recovering a detailed circuit diagram. They want to diagram the circuits of the brain? So it's a unique point in history where we have the right tools, techniques, and technologies for the first time ever to reveal the wiring diagram of the brain at the level of every single neuron and every single synapse. And this is Vogelsteen. Vogelsteen is a guy at IARPA talking about the Micron program. Each team plans to record the brain's roadmap differently. Cox's team will use a technique called two-photon microscopy to measure brain activity in rats as they are trained to recognize objects on a computer screen. The researchers will introduce a modified fluorescent protein, which is sensitive to calcium, into the rodents. Let's think about that for a second. The researchers will introduce a modified fluorescent protein, which is sensitive to calcium, into the rodents. Now, how would that happen? How is that possible? I'll show you. When a neuron fires, calcium ions rush into the cell, causing the protein to glow brighter. So, using a laser scanning microscope, the researchers will be able to watch the neurons as they're firing. That's a little bit like wiretapping the brain, Cox says. The way you might listen on a phone call to see what's going on, we can listen in on important internal aspects of the brain while the animal is alive and doing something. Then 1 cubic millimeter of the rat's brain will be sent to Jeffrey Lichtman, a biologist and neuroscientist also at Harvard. In Lichtman's lab, it will be cut into incredibly thin slices and imaged under a state-of-the-art electron microscope at enough resolution to see all the wire-like extensions of the brain cells that connect to each other. Tolias's team is taking a similar approach called three-photon microscopy to look into deeper layers of the mouse's brain and not just the top layers examined by Cox and his colleagues. Meanwhile, Lee's team plans to take a far more radical path towards mapping the connectome. Partnered with George Church, who we looked at the other day, and we'll continue to look at his work and his institutional, the laboratory at his institution, Harvard. A geneticist at Harvard Medical School, they plan to use DNA barcoding. George Church, a geneticist at Harvard Medical School, plans to use DNA barcoding. They will label every neuron with a unique sequence of nucleotides, which is what DNA is made of, aka a barcode, and chemically connect barcodes across synapses to reconstruct circuits. Just let that sink in for a minute. They're talking about barcoding your brain. Or a brain, excuse me. We're just dealing with rats here, right? But this is just what's public, right? Let's continue. While this method would not provide the same level of spatial information as microscopy, Lee hopes it will be faster and more accurate, provided it works as well at all. This technology has never been successfully used before, but if this barcoding technology works, it will revolutionize neuroscience and connectomics, Lee says. We are going to look at some of these technologies today, folks, just as soon as we get through this article. And all that constitutes the first half, and all that only constitutes the first half of the Microns project. The scientists next have to find a way to make all this information useful for algorithms in machine learning. They have some ideas about how to do this. For one, many researchers believe the brain is Bayesian, that neurons represent sensory information in the form of probability distributions. Sounds kind of quantumish. Calculating the most likely interpretation of an event based on previous experience. This hypothesis is based primarily on the idea of feedback loops in the brain. That information does not only flow forward, but that there are even more connections flowing back. In other words, researchers hypothesize that perception is not simply a mapping from some input to output, Rather, a constructive process, analysis by synthesis, exists in which the brain maintains and creates an internal representation of the world, generating expectation and prediction that allows it to explain incoming data and plan how to use it. This is a guiding principle we're looking at very closely. The hallmarks of this synthetic process, Cox says, we are where we confabulate what might be in the world and test that against what we actually see, and use that to drive our perception. For instance, the retina, which reacts to light by generating electrical impulses that are relayed to the optic nerve, and then the brain, is actually a two-dimensional structure. So when a person sees an object, perhaps the brain uses such a probabilistic model to infer a three-dimensional world from the light hitting the retina's two-dimensional surface. If that's the case, though, then the brain has found a much better way of approximating and inferring variables that we are capable of with our current set of mathematical models. After all, if you're observing a scene with 100 objects, consider only the forward and backward orientations objects may have, just two of the many. That's 2,100 possible patterns right there. Getting answers by computing all those probabilities is not feasible, and yet the brain does it effortlessly with an infinite number of possible orientations, different distances, with different rotations, in different lighting conditions. What the brain does is unfold this manifold of data points and make it easily separable, Tolias explains. Each of the three teams have recruited computer scientists to distill theories into models. Oh, where have we heard that before? Models. Well, the models say, anyway, shout out to Fauci and Birx if you're listening, which they will then test against the reverse engineered brain data. For any given description of an algorithm, such as a probabilistic algorithm, there are millions of implementation choices you have to make to translate that theory into code that executes, Vogelstein says. Of those million or so options, some combinations of those parameters and features will result in good algorithms, and some combinations will result in insufficient or bad algorithms. We're almost done with this article, folks. By extracting those parameters, settings from the brain, as opposed to guessing at them in software, as we have been doing, we will hopefully narrow the space down to a small set of implementations consistent with the brain. With such internal models, Microns, which is part of IARPA, an offshoot of DARPA, plans to make machines more automatic, particularly when it comes to training machines to identify objects without first having to run through thousands of examples in which the items are identified by name. Vogelstein wants to apply unsupervised learning techniques to aid U.S. intelligence. What could go wrong with that? We may have just a single picture or a single example of a cyber attack we want to prevent, or a single record of a financial crash or a weather event that causes a problem, he says. And we need to generalize a broader range of circumstances in which the same pattern might arise. So that's what we hope to achieve. Oh, is that all? You're just going to help with the weather? Sure. Better generalization, better capacity for abstraction, better use of sparse data. While the researchers agree that deriving such algorithms from the brain will be the most difficult part of Microns, they will have to determine a way to code how the brain processes information and forms new connections. Several challenges persist even in the earlier stages of the project. For one, their measurements of the brain will generate approximately 2 petabytes of data, the equivalent of the memory on 250,000 laptops or 2.5 million CDs. Storage of such a large data set will be difficult, and IARPA has partnered with Amazon to help find solutions. Oh, isn't that nice? Moreover, that data is all images. Mining it for information will require a process called segmentation, in which the structural elements of neurons and their connections are each colored differently so that the computer can make better sense of shared characteristics and patterns. Even if the whole world was coloring in for you, Lichtman said, it would take a lifetime to get the whole cubic millimeter colored in. Keep in mind, folks, they're still talking about just a cubic millimeter of a brain, not the entire thing. Instead, researchers will work on creating more sophisticated computer vision techniques to segment the data. Lichtman has already seen successes with a 100 terabyte data set, 1/20th the size of that which Microns plans to collect, generated from a piece of thalamus, that's part of the brain, a relay point for sensory information. His team's results will be published this month in Cell. We've learned that sometimes the same axons jump from one cell to another to contact the same place on different nerve cells, suggesting that the thalamus was organized differently than people expected. So what they're saying is that brains are dynamic, brain cells are dynamic, and they're not linear. Perhaps these results will extend to the cubic millimeter of cortex they have just started to assess. We know we can do large volumes, but now we're going to do what we call gigantic volumes. That's a huge step up. We think we're ready to go to this step. All right. So basically, it's a two-part process, right? Understand the brain biologically down to each wire, metaphysical wire, right? Metaphorical wire, each cell, each subcomponent of the cell, understand that part, the biology part, and then translate how it works into algorithms so that AI can have a human-like brain. That's what the goal of this project is, called Microns. Yeah, we're half hour in. We're moving on. So this is IARPA's Micron website. And we'll go through this. I'll show you this real quick. This is IARPA's homepage, iarpa.gov, IARPA.gov. And you can search for all kinds of cool stuff here. You can look at their programs, and it gives you a list of their programs. And some of these are really amazing. Kind of scary if you freak out, but look at their names: Odin, Mosaic, Raven, SMART, remote sensing, imaging, image processing, atmospheric correction, machine learning, radiometric calibration. This is their past research: Athena, Babel, Fuse, Mercury, Metaphor, natural language processing, cognitive science, cognitive linguistics, SHARP, cognition, psychometrics, fluid reasoning and intelligence, neuroscience, human performance. This is the intelligence agency's version of DARPA, and it's related to information, right? Intelligence requires information. Artificial intelligence would be a tool set to collect information. So let's go back to Microns. We'll spend more time on IARPA, as time permits. Micron seeks to revolutionize machine learning by reverse engineering the algorithms of the brain. The program is expressly designed as a dialogue between data science and neuroscience. Participants in the program will have the unique opportunity to pose biological questions with the greatest potential to advance theories of neural computation and obtain answers through carefully planned experimentation and data analysis. Over the course of the program, participants will use their improving understanding of the representations, transformations, and learning rules employed by the brain to create ever more capable neurally derived machine learning algorithms. Ultimate computational goals for Microns includes the ability to perform complex information processing tasks such as one-shot learning, unsupervised clustering, scene parsing, aiming towards human-like proficiency. So here's a PDF, Machine Intelligence from Cortical Network, Microns. And it's a diagram that breaks down biology and the idea of fusing it with artificial neural networks and how this would translate and how it work. Object identification, shape, color, distance, depth. We're not going to spend too much time on this, but this is an interesting infographic. Here is a breakdown of what something at very high resolution would look like from the brain. And here's some data points that are modeled after this. And we're going to start looking at some of their measurement tools and set the table for genetic manipulation, and then DNA nanotechnology, and then synthetic or metallic nanotechnology. That's where we're going over the next few episodes. And we're going to put all this stuff together. But I feel that it's important to show you how they look at this level of resolution before I show you what they're looking at and what they're trying to do at this resolution. So that's what we're going to focus on tonight. Realist Anon, I see you out there. It's 2-hour blocks of time, and it's Sunday night, Monday morning, depending upon where you are. So East Coast time, it's going to be 1 a.m. to 3 a.m. And then on Friday mornings, East Coast time, it's 2 a.m. to 4 a.m. And the show page needs to be updated, but we just changed the time slot for Sunday. So if you go to the show page on Patriot Soapbox, you'll see there's a calendar here, show calendar. And this is the best place to go. This is updated the most frequently. And this show calendar will give you the times of shows. And you can correlate the show calendar with the shows. And if you ever miss a show, what you can do is you just go to the shows page on patriotsoapbox.com, find the show you want. In this case, it's Big Tech. And the most recent shows are actually posted to the page, but you can view them all with the hyperlink here, and it takes you to Patriot Soapbox archive channel, where all of the shows since the beginning of this show, for example, have been archived. And we're on episode 33. So the first show is going to be at the bottom, and the most recent show is going to be at the top. And once this one's over, then it'll also be archived here. It's a good question. Thank you for asking. So here we go. We are going to go to the Wyss Institute. And what I did is I searched in a term for paint. And the reason why is because it's a term that Wyss has used, and they paint DNA, right? That's just a fun way for them to say that. So I encourage you to go to harvard.edu, find the Wyss Institute. You can type in the Wyss, W-Y-S-S Institute, and there's a search box up here, and you can type in things. Once you kind of browse the website, you can find terms that are of interest to you, and I'll give you an example. This is Exchange Paint, Neurons Up Close and Personal, and there are tags over here, and one of the tags is DNA paint. There's super resolution microscopy, which we're going to look at because we read in that article that they were down to 2-photon, 3-photon microscopy. This is electron microscopes, but much more improved, much higher resolution. Nanotechnology, molecular robotics, DNA nanostructures, this kind of stuff we're going to be looking at over the next few shows. So we're going to look at neurons up close and personal first, and we'll continue. So there's no sound here. We will come back probably and look at this again. But what this is a slice. And all the different colors and the different lines, they have painted DNA. So DNA comes in strands, and they are able to mark and code and understand relationships between the DNA and or the connections that are made. You can see here, they're funny names on the top. These are different protein types, and they're able to fluoresce them and dye them specific colors, and then they can image them. And you can overlay this stuff, especially with big data. You can overlay this stuff after you've recorded like neurons firing, the neuron activity, and you can overlay it and see what's working when and how, and start mapping the circuitry of the brain. So they're able to go in and genetically modify brains to express fluorescent proteins so that when a particular DNA structure that they're looking for activates or fires or does something, then it illuminates in a color, and they can color code this stuff in your biology. And this is likely something from a mouse or a rodent. but they can culture human tissue. We'll look at some of that too. Brain on a chip. If we get that far tonight, we'll see how far we get. All right. So this is inexpensive super resolution microscopy. So we are going to look at some of their toolkits, their tool sets, before we get into what they can do at this level. I want to illustrate for you in as great a detail as I can that it's possible for them to work at the nanometer scale with surgical precision at this point. Accurate and precise microscopic imaging platform for visualizing different molecular species in biological samples. Wyss Institute scientists have developed a highly versatile and inexpensive microscopic imaging platform designed to visualize objects with molecular scale resolution and unprecedented complexity. The DNA-powered imaging technology can reveal the inner workings of cells at the single molecule level using conventional microscopes found in most laboratories. Key to the Wyss Institute's DNA-driven imaging super resolution technology is the interaction of two short strands of DNA, one called the docking strand that is attached to the molecular target to be visualized, and the other called the imager strand, which carries a light-emitting dye. Despite the recent revolution of optical imaging technologies that has enabled the distinction of molecular targets residing less than 200 nanometers apart from each other, modern super resolution techniques have still been unable to accurately and precisely count the number of biomolecules at cellular locations. Okay, modern super resolution techniques have still been unable to accurately and precisely count the number of biomolecules at cellular locations. With DNA paint, super resolution microscopy, and Q-paint analysis, researchers will be able to quantify the number of molecules at specific locations in the cell without the need to spatially resolve them, because the paint will do it for you, without requiring an expensive, large super resolution microscope. Okay. We aim to provide an ultimate view into biology. Quote by Pen Yin. Ultiview Inc., a startup launched out of the Wyss Institute, is focused on developing imaging solutions to unfold new areas of biology by leveraging DNA paint to enable the precise co-localization of an unlimited number of targets. So this is not exclusive to the Wyss Institute. They have created a product for sale for other research facilities to use to perform this type of imaging. Here we go.
Biological systems are fundamentally complex molecular systems. It has been challenging to visualize biology on the molecular scale.
Biological systems, it's been challenging to visualize them on the biological scale.
Especially for single molecules in crowded molecular environments.
Especially single molecules in crowded environments.
Discrete molecular imaging technology, or DMI, enable us to visualize biology using optical microscope on the 5 nanometer scale.
Okay, so optical resolution to 5 nanometers. 5 nanometers.
5 nanometer is not a random number. 5 nanometer is the size of a small protein. This kind of resolution is the highest resolution for visualizing molecular components.
We named our technology discrete molecular imaging because before we were only able to look at a big protein complex as a cloud, and now we're able to look at them as individual protein molecules. We used a novel combination of DNA nanotechnology and computational methods to really pin these molecules in space so that we can clearly visualize each of them.
Okay, so 5 nanometers, you're down to molecules, you're looking at specific proteins. The idea here is to understand the brain as specifically and as in a targeted way as possible so that you can create a circuit diagram, a wiring diagram of the brain. And that's done by imaging DNA, because DNA ultimately creates proteins and or other components of a cell, like a neuron.
We developed the discrete molecular imaging based on our previous DNA paint technology for super resolution imaging. DNA paint uses transient binding between two complementary DNA molecules, one that is attached to the molecular target of interest, one that is floating in the solution and conjugated to a fluorescent dye. When they bind together, they create transient blinking events at the molecular target site that are visible through the microscope to create super resolution microscopy images. And in this platform, we already have exchange paint technology that allows for multiplexed imaging and simultaneous observation of different type of protein or molecular features. And also,
so it's not just one color at a time, folks. They can introduce many genetic variations into a sample. We'll keep this as objective as possible. And they can code these insertions to look for specific pieces of DNA, because components of the cell will have specific protein expressions, which means they have specific genetic code. They can look for specific pieces of DNA and fluoresce or bind to many different locations simultaneously to ultimately create more than one color in an image.
So QPaint, which stands for quantitative paint, that allows researchers to quantitatively count the number of molecular features within a big complex. Now with DMI, we're adding the extra power of being able to resolve every single molecular feature that are on the protein size, which is about 5 nanometer in size.
So in this work, we have demonstrated DMI using synthetic targets. Now moving forward, we are very excited to apply this method to visualize real biological samples.
Protein-protein interactions are the foundations of biological life. Visualizing them in the context is only the first step in trying to potentially manipulate them on a single molecule level.
Oh, first we have to know where they are, and then bind that or tie that into what they do, and then they can begin to manipulate what they do. Manipulating protein expressions at the molecular/cellular level. That's what we're made of, folks. That's what biology is made of.
Moving forward, we think about DMI will enable transformative insights into biology and medicine. We are very excited about this development, and we believe DMI is an exciting step forward towards obtaining an ultimate view of biology.
Hazel, yeah, it's related to Dr. Lieber's work. The heads of the departments all talk to each other, and George Church and Lieber, I'm sure, knew each other. The patents that Dr. Lieber has discuss fusing his nanotechnology, which are nanoelectronics primarily, into tissue. And in order to do that intelligently, you would have to know how to target tissue, which is why we're getting into the genetics piece. So if you can selectively target tissue by decoding and understanding the genetics, you can then begin to selectively apply nanoelectronics. But it gets crazier than that. If they can manipulate genetics, then they can just tell the cells to grow or produce certain alterations so that electronics aren't needed for a computer interface. They can have cells express more magnetic material like iron, ferritin, for example, and have it be produced and then strategically placed so you can grow your own magnetic interface with biology through genetic modification. This can also be done with light, cells that respond to light. That's known as optogenetics. The magnetic piece I was referring to is known as magnetogenetics, and we will cover that. And for those of you that want a head start into that, it is in the show notes, but it is beyond the scope of our discussion today. Well, here's some of the nanotech, CRISPR gene modification, which we'll discuss next likely. Here's nanobiotechnology. This is all in the show notes. These will be in upcoming shows. For those of you that want a head start, have at it. You can build with DNA. Thumper, you there?
Yes. Hello.
Hi. I lost. I don't see anything shared on chat. It looks like I got kicked off.
I got kicked off too.
Interesting. All right. Bear with me, folks. We'll bring chat back up, or we'll bring the share back up and see if it shows up. Yes. Okay, cool. So chemogenetics, designer drugs, right? Designer pharmaceuticals, they can genetically modify cells so that they are more effective with the designer pharmaceuticals. And then optogenetics. I'm going to pull more information for optogenetics, but that's where they can change the cells to respond to light. So it's all in the show prep. We're just not there. We're not going to cover that quite yet, but we will. So let's get back to microscopy. So Purdue, new development in 3D super resolution imaging. Awesome. Thanks, chat. Thank you, chat. I got your 5x5s. Very good. Purdue, recent studies show that 40% of Americans over the age of 85 have Alzheimer's disease. Right? So this is just for medical use to make you better. And that the disease begins 10 to 20 years before people show up at the doctor's office with memory problems. One major problem with understanding Alzheimer's is not being able to clearly see why the diseases start. A super resolution nanoscope developed by Purdue University, researchers now provide a 3D view of brain molecules with 10 times greater detail. This imaging technique could help reveal how diseases progress and where new treatments could intervene. The instrument helped Indiana University researchers better understand the structure of plaques that form in the brain of Alzheimer's patients, pinpointing the new characteristics that are possibly responsible for damage. Published findings appear in the journal Nature Methods. Long before Alzheimer's develops in an individual, waxy deposits called amyloid plaques accumulate in the brain. These clusters interact with surrounding cells, causing inflammation that destroy neurons and create memory problems. The deposition of these plaques is currently the earliest detectable evidence of pathological change leading to Alzheimer's disease. While strictly a research tool for the foreseeable future, the technology has allowed us to see how plaques are assembled and remodeled during the disease process, says Gary Landreth, professor of anatomy and cell biology at the Indiana University School of Medicine's Stark Neuroscience Research Institute. It gives insight into the biological causes of the disease so that we can see if we can stop the formation of these damaging structures in the brain. The limited resolution in conventional light microscopes and the natural thickness of the brain tissue have prevented researchers from clearly observing 3D morphology of amyloid plaques and their interactions with other cells. Brain tissue is particularly challenging for single molecule super resolution imaging because it's highly packed with extracellular and intracellular constituents with distort and scatter light, our source of molecular information, said Fang Huang, Purdue Assistant Professor of Biomedical Engineering. You can imagine deep into the tissue, but the image is blurry. The super resolution nanoscopes, which Huang's research team has already developed to visualize cells, bacteria, and viruses in fine detail, uses adaptable optics, deformable mirrors that change shape to compensate for light distortion called aberration that happens when light signals from single molecules travel through different parts of a cell or tissue structured at different speeds. We're going to learn about a lady named Mary Lou Jepson, who has a company by the name of Open Water, who deals with interferomic holography so that they can read your brain using infrared light without going into your brain. Same general idea. What we're talking about here is complex optics, right? With the use of computers and machine learning to correct for light dispersion, light refraction, light reflection, and understanding those characteristics based on tissue so that a clean image can be viewed or rendered once the light is at the receive point. To tackle the challenge of brain tissue, Huang's research team developed new techniques that adjust the mirrors in response to sample depths to compensate for aberrations introduced by the tissue. At the same time, these techniques intentionally introduce extra aberration to maintain the position information carried by a single molecule. The nanoscope reconstructs the whole tissue, its cells, and the cell constituents at a resolution 6 to 10 times higher than conventional microscopes, allowing a clear view through 30 micron thick brain section of a mouse's frontal cortex. The researchers used mice that were genetically engineered to develop the characteristic plaques that typify Alzheimer's disease. All right. Let's see.
My team works on developing high-resolution microscopy methods by detecting single molecules. Brain tissues are particularly challenging for single molecule imaging because it's packed with extracellular and cellular structures. So we develop an algorithm to efficiently drive the deformed mirror to form different shapes, and the shape is used to compensate the distortion. And also we introduce extra distortions such that to allow this point spread function or the detected pattern to maintain its information content through different tissue depths.
Pretty cool. It's a very, very high-powered microscope that has AI, basically, machine learning, algorithms, whatever you want to call it, some version of automation that's driving mirrors and light emissions out of this microscope in such a way so that they can render very, very clear images of things that are very, very small.
Now, in Alzheimer's disease, one of the most prominent pathological features is the accumulation of what's called amyloid plaques. We are investigating at a very fine level the structure of these plaques to give us insight into the underlying biological processes by which they're generated.
This system is a three-dimensional super-resolution microscope that allows you to look deep into the tissue while at the same time achieving 6 to 10 times higher resolution than conventional microscopes.
That is remarkable, in my view. I mean, the science and technology is phenomenal, folks. Just how is it being used? If we can give quality of life to elderly patients as they're advertising here, great. But through this quest, right, what the sales pitch that we all refer to it as, the intricacies of the brain are being understood. And as we get into the methods in which genetic modification can occur and some of the things that can be done, we'll start putting these pieces together to ultimately illustrate the fact that we're going to be integrated into the internet. It's going to happen. And we need to understand how and understand that we need to demand accountability. We need to demand accountability.
From our perspective, this is really important because we are able to see things we have never been able to see before and gives us insight into the biology.
This method potentially could allow us to further our understanding of the mechanism of these diseases and potentially to find the cause of those.
The reason why this is important is that in the last few years, we've come to understand that the beginning of Alzheimer's disease occurs 10 to 20 years in advance of people showing up at their doctor's office being forgetful and having memory problems. This fundamentally changes how we think about treating the disease, because if it starts 2 decades before dementia occurs, that will force us to treatment paradigms to treat essentially younger people.
It is very exciting to see these two teams from Purdue and IU work together extensively.
You know how much cheaper medicine will be if the tools become good enough to identify issues years before they become a symptom, a problem? You know what the likelihood of a cure is at that point? But we have to get past this idea of symptom management and start getting to an idea or a concept of curing. Our medical system is in the business of symptom management, not in cures. They'll sell it to you like they're looking for cures, but the reality is that the care that people like you and I get? We get symptom management so they can soak our money.
To develop something that is unique for Alzheimer's studies.
Ah, what lovely, pleasant music. Let's continue. Kurzweil. Here we go. Expanding the brain achieves super resolution with ordinary confocal microscopes. We're about an hour in. We're doing pretty good. What does this mean? Ordinary confocal microscopes, so existing proliferated microscopes. Engineers at the MIT-based Center for Brains, Minds, and Machines, the MIT-based Center for Brains, Minds, and Machines. Now, let's break that down a little bit. Brain, the physical matter that is what we understand as a brain. The mind. Now, they made the distinction in the title of this school or this center at this prestigious school. So the mind being different than the brain. We could encapsulate that as the processor that the gentleman from MIT spoke of earlier, Mr. Edward Boyden, or the consciousness, right? Or the interface between the hardware and the spirit, so software, the mind, when we compare it to a brain versus a mind, right? And then you have machines, and this is one center under which they all are formed. Brain, mind, and machines from MIT have developed a way to make a brain expand to about 4.5 times its usual size, allowing nanoscale structures to appear sharp with an ordinary confocal microscope. The new expansion microscopy technique uses an expandable polymer and water to enable researchers to achieve super resolution to resolve details down to about 70 nanometers laterally compared to about 300 nanometers, that's the violet light, which was the previous limit with a diffraction-limited conventional microscope, and without the slower performance of existing super resolution microscopes. So the 5 nanometer microscope we were looking at, super resolution microscopes, it says here they perform slower, right? Probably a lot more processing going on. The conventional microscopes don't have as good a resolution, 70 nanometers, right? Instead of 5, but it's much faster. So they each have their place in this investigation as it relates to unpacking and decoding the circuit diagram within the brain. Here's some images. Shown here are 3D images of a mouse brain tissue taken using super resolution microscopy, a technique that uses fluorescent molecules to resolve tiny brain details of a portion of the hippocampus, showing neurons green and synapses blue and red. Credit Ed Boyden, Fei Chen, and Paul Tilburg. Expansion microscopy may provide a key tool for comprehensive, precise, circuit-wide brain mapping, Boyden said. The team has demonstrated the process on a mouse, fruit fly, and zebrafish brains, and is working with another team to apply it to human tissue. Dendrites, tree-like extensions of the neuron cell body, are the primary sites for receiving and integrating information from other neurons. This is brain cells. These are brain cells, folks. This 3D animation of dendrites in a mouse brain was taken using super resolution imaging. Boyden adds that the process may be useful beyond the brain to other parts of the body. Many types of biological processes involve nanoscale interactions across large systems, such as cancer metastasis and immunological responses. This clever technique which bypasses the limitations of traditional techniques for brain imaging, has the potential to be a powerful new tool to help researchers map and understand the brain. Says so-and-so from so-and-so. Pramod, not even going to try to pronounce the last name, NSF Assistant Director for the Engineering Directorate, which supports an array of neuroengineering projects. Another 3D animation of dendrites in a mouse brain, taken using super resolution microscopy. Pretty amazing the resolution that they can get to. The new method is another advance in brain imaging that brings researchers closer to illuminating the entire brain and nervous system. One of today's greatest engineering challenges. Development of expansion microscopy was detailed in the January 15th issue of Science. The Center for Brains, Minds, and Machines is an NSF Science and Technology Center funded in 2013 as part of NSF's continuing support for the advancement of fundamental brain research. All right. We're going to watch some of this and then we'll continue. We have time. Good.
Hello, everybody. I brought with me today a baby diaper. You'll see why in a second. Baby diapers have interesting properties. They can swell enormously when you add water to them, an experiment done by millions of kids every day. But
oh, it gets better.
The reason why is that they're designed in a very clever way. They're made out of a thing called a swellable material. It's a special kind of material that when you add water, it will swell up enormously, maybe 1,000 times in volume. And this is a very useful industrial kind of polymer. But what we're trying to do in my group at MIT is to figure out if we can do something similar to the brain. Can we make it bigger, big enough that you can peer inside and see all the tiny building blocks, the biomolecules, how they're organized in 3 dimensions, the structure, the ground truth structure of the brain, if you will.
If you will. So crazy, right? We're going to go back here to microns, revolutionizing machine learning by reverse engineering algorithms of the brain. And this article discussed the 2 parts required. Break it down at the cellular, subcellular level, and then translate that into software. Let's continue.
If we could get that, maybe we could have a better understanding of how the brain is organized to yield thoughts and emotions and actions and sensations. Maybe we could try to pinpoint the exact changes in the brain that result in diseases like Alzheimer's and epilepsy and Parkinson's, for which there are few treatments, much less cures, and for which very often we don't know the cause or the origins and what's really causing it to occur.
Now, for our 10-second pause. We are going to watch some more of this and then probably skip some and then continue.
Our group at MIT is trying to take sort of a different sort of point of view from the way that neuroscience has been done over the last 100 years. We're designers, we're inventors, we're trying to figure out how to build technologies that let us look at and repair the brain. And the reason is the brain is incredibly, incredibly complicated. So what we learned over the first century of neuroscience is that the brain is a very complicated network made of very specialized cells called neurons with very complex geometries.
Look familiar, right? Look familiar? Kind of similar, yeah? All right, here we go.
And electrical currents will flow through these complexly shaped neurons. Furthermore, neurons are connected in networks. They're connected by little junctions called synapses that exchange chemicals and allow the neurons to talk to each other. Now, the density of the brain is incredible. In a cubic millimeter of your brain, there are about 100,000 of these neurons and maybe 1 billion of those connections.
Gee, where have we heard that measurement before? That's exactly what IARPA's looking for in their project called microns.
But
does anyone know what a micron is?
it's worse. So if you could zoom in into a neuron, and of course this is just our artist rendition of it, what you would see are thousands and thousands of kinds of biomolecules.
Okay, hope you're paying attention. This is where it gets pretty interesting, folks. Thousands and thousands of biomolecules in a neuron. Each one of the biomolecules, right, has a unique signature. Just remember this, because we're going to get into genetic engineering here shortly, probably next episode. So Friday morning. Just thousands of biomolecules. Let's back this up just a touch.
So if you could zoom in into a neuron, and of course this is just our artist rendition of it, what you would see are thousands and thousands of kinds of biomolecules, little nanoscale machines that are organized in complex 3D patterns. And together, they mediate those electrical pulses, those chemical exchanges that allow neurons to work together to generate things like thoughts and feelings and so forth.
Okay. So you have the macro structure on the left, which is really not very big at all, right? A synapse, a neuron, a piece of a neuron, neurons in the background. And then you have the subcomponents, the biomolecules of a neuron. They want to, in this program, understand down to each one of these biomolecules, what it does, why it's configured the way it is, when it activates or performs its function, what happens? How are these built? When are they replaced? How are they expressed? Down to the genetics of it. Everything about it in this program, so that they can take all of that collective knowledge and translate it into artificial intelligence. But that's only one piece. Once they understand the biological structures, what they do, now they can target them for things like gene therapy. And we'll get into some technologies that relate to gene therapy that allow cells to interact with energetic sources like electromagnetic fields, light, perhaps even sound, chemicals. Sort of highlighted that a little bit ago before the share crashed right at the same time we were highlighting it, but let's continue.
Now, we don't know how the neurons in the brain are organized to form networks, and we don't know how the biomolecules are organized within neurons to form these complex organized machines. If we really want to understand this, we're going to need new technologies. But if we could get such maps, if we could look at the organization of molecules in neurons and neurons in networks, maybe we could really understand how the brain conducts information from sensory regions, mixes it with emotion and feeling, and generates our decisions and actions. Maybe we could pinpoint the exact set of molecular changes that occur in a brain disorder. And once we know how those molecules have changed, whether they've increased in number or changed in pattern, we could use those as targets for new drugs, for new ways of delivering energy into the brain in order to repair the brain computations that are afflicted.
Well, goodness gracious, I think he just said what? Well, do we want to replay that? Right? Understand the biomolecules to be able to genetically target, and then also understand them in such a way so that we can begin to introduce energy selectively into the brain for therapy. This guy is the head of his field. He is by trade an optogeneticist, but well-versed in many other technologies, a neuroscientist and a neural engineer. That is what this gentleman does with his time as a professor and inventor at MIT.
In patients who suffer from brain disorders. We've all seen lots of different technologies over the last century to try to confront this. I think we've all seen brain scans taken using MRI machines. So these, of course, have the great power that they are non-invasive. They can be used on living human subjects. but also they're spatially crude. Each of these blobs that you see, or voxels as they're called, can contain millions and millions of neurons. So it's not at the level of resolution where we can pinpoint the molecular changes that occur or the changes in the wiring of these networks that contributes to our ability to be conscious and powerful beings.
Oh, well, I bet there's a solution, Mr. Boyden, and I bet you're going to tell us one of the ways to get down to the neural circuitry, the neural wiring.
At the other extreme, you have microscopes. So microscopes, of course, will use light to look at little tiny things. And for centuries, they've been used to look at things like bacteria. For neuroscience, microscopes are actually how neurons were discovered in the first place, about 130 years ago. But light is fundamentally limited. You can't see individual molecules with a regular old microscope. You can't look at these tiny connections. So if we want to make our ability to see the brain more powerful, to get down to that ground truth structure, we're going to need to have even better technologies. So in my group, a couple years ago, we started thinking, why don't we do the opposite? If it's so darn complicated to zoom into the brain, why can't we make the brain bigger? Initially started with 2 graduate students in my group, Fei Chen and Paul Tilburg, and now many others in my group are helping with this process.
Okay. We'll keep playing. I don't want to cut myself short for the rest of what I want to show you, but this is kind of important.
We decided to try to figure out if we could take polymers, like the stuff from the baby diaper, and install it physically within the brain. If we could do it just right, and you add water, you could potentially blow the brain up to the point where you could actually distinguish those tiny biomolecules from each other. You could see those connections and get maps of the brain. This could potentially be quite dramatic. And so we brought a little demo here. We got some purified baby diaper material. It's much easier just to buy it off the internet than to extract the few grains that actually occur in these diapers. And I'm going to put...
All right, so we'll watch it expand as he pours water.
Of the baby diaper material can increase in size. And what you're going to see is it's going to increase in volume by about 1,000-fold before your very eyes.
So the benefit to this is that it expands uniformly, up and out and around uniformly.
If you could zoom in, it might look something like what you see on the screen. Polymers are chains of atoms arranged in long, thin lines. And the chains are very tiny, about the width of a biomolecule, and these polymers are really dense. They're separated by distances that are around the size of a biomolecule. So this is very good because we potentially can move everything apart in the brain. If we add water, what will happen is this swellable material is going to absorb the water, the polymer chains are going to move apart from each other, and the entire material is going to become bigger. And because these chains are so tiny and spaced by biomolecular distances, we can potentially blow up the brain and make it big enough to see. So here's the mystery then. How do we actually make these polymer chains inside the brain so we can move all the biomolecules apart?
That's a good question. Self-assembly?
If we could do that, maybe we could get these ground truth maps of the brain. We could look at the wiring. We can peer inside and see the molecules within. So to explain this, we made some animations where we actually look at, in these artist renderings, what biomolecules might look like and how we might separate them. Step 1. What we have to do, first of all, is to attach to every biomolecule, shown in brown here, a little anchor, a little handle. We need to pull the molecules of the brain apart from each other, and to do that, we need to have a little handle that allows those polymers to bind to them and to exert their force.
So at this level, that would require some kind of an attachment, maybe chemical or a genetic attachment. Right? So you're, like you said, a handle, some kind of a mount, like for a joist in a house or something, some kind of a mount. Keep in mind that in this example, these are not live samples, right? This would be something in a dish that would be like a thin slice of a brain to then blow it up to understand the circuitry. But we'll just let your minds wander, folks, because if you can think of it, you better believe these people have thought of what could be done, right? So let's continue.
Now, if you just take baby diaper polymer and dump it on the brain, obviously it's just going to sit there on top. So we need to find a way to make the polymers inside. And this is where we're really lucky. It turns out that you can actually get the building blocks, monomers as they're called. And if you let them go into the brain and then trigger the chemical reactions, you can get them to form those long chains right there inside the brain tissue. They're going to wind their way around biomolecules and between biomolecules, forming those complex webs that will allow you eventually to pull apart the molecules from each other.
Okay. Well, if they can self-assemble like that in a known way, something that's preferable, well, now all you got to do is code those things to attach little nanoparticles of whatever, and you've created an electrical mesh in the brain. But anyway, this is just a brain slice, and it's for science, and it's for Alzheimer's disease and whatever else they're going to talk about, epilepsy and blah blah blah. So, these people love you. Let's continue.
And every time that one of those little handles is around, the polymer is going to bind to the handle, and that's exactly what we need in order to pull the molecules apart from each other. All right, the moment of truth. We have to treat this specimen with a chemical to kind of loosen up all the molecules from each other, and then when we add water, that swellable material is going to start absorbing the water. The polymer chains will move apart, but now the biomolecules are going to come along for the ride. And much like drawing a picture on a balloon, and then you blow up the balloon, the image is the same, but the ink particles have moved away from each other. And that's what we've been able to do now, but in 3 dimensions.
So, great analogy, right? A balloon, expand the balloon, draw a little happy face on the balloon. The happy face is still there, only it's a larger happy face when the balloon is inflated. Same general principle. Rottweiler, in this example, no, they're not talking about creating artificial pathways, but we're going to discuss those concepts as time goes on. The point of this extended illustration is to show you some techniques and observation methods in order to provide you with some initial visualizations so that when we get into the genetic manipulation and we get into some of these other technologies, you then have a conceptual understanding. You can visualize sort of where and how this stuff might be applied in biology. That's the purpose of going through this in this order. Great question, though.
There's one last trick. As you can see here, we color-coded all the biomolecules brown. That's because they all kind of look the same. Biomolecules are made out of the same atoms, but just in different orders. So we need one last thing in order to make them visible. We have to bring in little tags with glowing dyes that will distinguish them. So one kind of biomolecule might get a blue color, another kind of biomolecule might get a red color, and so forth. And that's the final step.
Yeah, that's called genetic modification. That's called find a receptor site that's specific to a particular biomolecule you're looking for, and then code some DNA or RNA, probably RNA, to then go in and look for its partner, like a lock and a key, finding the right lock, and then it will attach. And you can fluoresce and make different biomolecules show up. Case in point, very simple example here. You have neurons, fluorescent molecules to resolve tiny brain details of a portion of the hippocampus, showing neurons in green and synapses blue and red. These are just the overall larger metastructures, the synapses and the neurons. This guy's talking about going in at the neuron and breaking down or segmenting the biomolecules that make up the neuron. So these green things being the neuron, going into one of them, for example, and breaking up and expanding the biomolecules in one of them, the subcomponents of the neuron, and tagging those using synthetic biology, genetics through the process of synthetic biology. Genetic modification to tag specific biomolecules within a single neuron. I know I'm repeating myself, but I really want to drive this home with respect to the level of resolution, how fine they are able to view, visualize, and interact. Most importantly, interact. They're able to interact with the subcomponents of a cell, many of them within a single cell. using genetics. Notice the weird little double helix kind of a thing right here, right? For the visualization, that's what they're showing here, is a helix, RNA or DNA, to attach to this specific. So this is coded a specific way. So when it finds the right biomolecule, the right protein, then it makes its attachment. If it went over here, for example, or somewhere else, and it wasn't the right match, like a lock and a key, it wouldn't bind. So red would only go with something they intended to fluoresce red. Blue would only go with something they intended to fluoresce blue, based on their unique genetic codes for each of these biomolecules. This is surgical at this point. Absolutely surgical, down to the subcomponents of a neuron, the biomolecules in a neuron.
Now we can look at something like a brain and look at the individual molecules because we move them far apart enough from each other that we can tell them apart. So the hope here is we can make the invisible visible. We can turn things that might seem small and obscure and blow them up until they're like constellations of information about life. And so here's an actual video of what it might look like. We have here a little brain in a dish, a little piece of a brain actually, and we've infused the polymer in, and now we're adding water. And what you're going to see is that right before your eyes, this video is sped up about 60-fold. This little piece of brain tissue is going to grow, and it can increase by 100-fold or even more in volume. And the cool part is, because those polymers are so tiny, we're separating biomolecules evenly from each other. It's a smooth expansion. We're not losing the configuration of the information. We're just making it easier to see.
Critical. They're not losing the configuration, because remember, the idea here is to map the brain, to understand the neural circuitry. Let's search for wiring. Unique point in history where we have the right tools, techniques, and technologies for the first time to reveal the wiring diagram of the brain at the level of every single neuron and every single synapse. Let's continue. Wyss Institute will lead IARPA-funded brain mapping consortium. The Wyss Institute for Biologically Inspired Engineering at Harvard. Let's before we go any further, because we're jumping around between Harvard and MIT. So I'm going to show you just how interconnected they are. The Broad Institute of MIT and Harvard is empowering a revolution in biomedicine to accelerate at the pace at which the world conquers disease. So the Broad Institute is a partnership, right? It is a partnership between Harvard and MIT. Harvard University, California Institute of Technology, Harvard Medical School, IBM, MIT, Calico, Apple, Hospital, Office of Technology Services and Security, Commonwealth of Massachusetts, President of Massachusetts Institute of Technology. Oh, hi, Mr. Schmidt, former executive chairman of Alphabet. Nice to see you at the Broad Institute. Anyway, so it all flows back and forth. So let's continue. That was a new one for me, folks, seeing that gem of a fellow. The Wyss Institute, part of Harvard, for Biologically Inspired Engineering at Harvard University, today announced a cross-institutional consortium to map the brain's neural circuits with unprecedented fidelity. The consortium is made possible by a $21 million contract from the Intelligence Advanced Research Projects Activity, IARPA, and aims to discover the brain's learning rules and synaptic circuit design, further helping to advance neurally derived machine learning algorithms. The consortium will leverage the Wyss Institute's FISC-Q, fluorescent in situ sequencing, method to push forward neuronal connections, the science of identifying the neuronal cells that work together to bring about specific brain functions. The FISQ was developed in 2014 by the WISCOR faculty member George Church. Mr. Church. Epstein associate, I should say, and colleagues, and used, unlike traditional sequencing technologies, it provides a method to pinpoint the precise locations of specific RNA molecules in intact tissues. So this FISC or FISQ technology is able to pinpoint precise locations of specific RNA molecules down to the RNA in intact tissue. The consortium will harness the FISQ capability to accurately trace the complete set of neuronal cells and their connecting processes in intact brain tissue over long distances, which is currently difficult to do with other methods. Oh, wait, it gets better, folks. Awarded a competitive IARPA Microns contract, the consortium will further the overall goals of President Obama's BRAIN Initiative, which aims to improve the understanding of the human mind and uncover new ways to treat neuropathological disorders like Alzheimer's disease, schizophrenia, autism, and epilepsy. The consortium's work will fundamentally innovate the technological framework used to decipher the principal circuit neurons used to communicate and fulfill specific brain functions. The learnings can be applied to enhance artificial intelligence in different areas of machine learning, such as fraud detection, pattern and image recognition, and self-driving car decision-making. Wow. This has Apple and Tesla and Twitter and everybody all over it, doesn't it? Yes. Historically, the mapping of neuronal paths and circuits in the brain has required brain tissue to be sectioned and visualized by electron microscopy. Complete neurons and circuits are then reconstructed by aligning the individual electron microscope images. This process is costly and inaccurate due to the use of only one color, gray, said Church, who is the principal investigator for the IARPA Microns Consortium. Interesting, huh? Like the head dude. Head guru, Mr. Church. We are taking an entirely new approach to neuronal connectomics, immensely colorful barcodes that should overcome this obstacle. And by integrating molecular and physiological information, we are looking to render a high definition map of neuronal circuits dedicated first to specific sensations, and in the future to behaviors and cognitive tasks. Now, what are specific sensations, folks? Breath of fresh air? Pat on the back? Kiss on the cheek? Slap in the face? Let's continue. Church is the professor of genetics at Harvard Medical School and professor of health sciences and technology at Harvard and MIT. To map neural connections, the consortium will genetically engineer mice so that each neuron is barcoded throughout its entire structure with a unique RNA sequence, a technique called bionic barcoding of individual neuronal connections, developed by Anthony Zador at Cold Springs Harbor. For those of you that are not familiar with Cold Springs Harbor, you should do some research on Cold Springs Harbor. You might find affiliations with Bill Gates. But let's continue. Thus, a complete map representing the precise location, shape, and connections of all neurons can be generated. I will read this paragraph again without interrupting because this is very important. To map neural connections, the consortium will genetically engineer mice so that each neuron is barcoded throughout its entire structure with a unique RNA sequence, a technique called bionic barcoding of individual neuronal connections, developed by Anthony Zador at Cold Springs Harbor Laboratory. Thus, a complete map representing the precise location, shape, and connections of all neurons can be generated. The key to visualizing this complex map will be the FISQ which is able to sequence the total complement of barcodes and pinpoint their exact locations using a super resolution microscope. Importantly, since FISQ analysis can be applied to intact brain tissue, the error-prone brain sectioning procedure that is part of common mapping studies can be avoided, and long neuronal processes can be more accurately traced in larger numbers and at a faster pace. In addition, the scientists will provide the bar-coded mice with sensory stimulus, such as a flash of light, to highlight and glean the circuits corresponding to that stimulus within the much more complex neuronal map. Freaky, folks. An improved understanding of how neuronal circuits are composed and how they function over longer distances will ultimately allow the team to build new models for machine learning. Now, if those few paragraphs don't make the hair stand up on your arms or the back of your neck, I don't know what will, because that is alarming. Anyway, you know, the vaccine for COVID-19 is coming out soon. And, you know, the shot regimen for most children up to the age of 15 now, all the way up to 18, hundreds of shots. Yeah, I'm digressing. Let's get back to, the multidisciplinary consortium spans 6 institutions. In addition to Church, the Wyss Institute's effort will be led by Samuel Inverson, PhD, who is staff software engineer, so computer science, and co-investigator of the project. Complementing the Wyss team are co-principal investigators Anthony Zador, PhD, Alexei Kulika, PhD, Jay Lee, PhD, at Cold Springs Harbor Laboratory, where you can get your lobotomy at the laboratory. Adam Marblestone, PhD, and Liam Piniski, a PhD, are co-investigator at MIT and co-principal investigator at Columbia University, respectively. The Harvard-led consortium is partnering with another Microns team led by Taising Li, PhD, of Carnegie Mellon University, as principal investigator under a separate multi-million-dollar contract, with Sandra Kuhlman, PhD, of Carnegie Mellon University, and Alan Yu Lee, PhD, of Johns Hopkins University, as co-principal investigators to develop computational models of the neural circuits and the new generation of machine learning algorithms by studying the behaviors of large populations of neurons in behaving animals. In behaving animals. Oh, do behave, good animals. Do behave. What that means is they're doing this in live specimens. They are alive, and their brains are barcoded, and they can map the brains with the barcodes. MKOTB, and I'm not referring to NKOTB. I'm referring to MKOTB. MOKTB, mark of the... I've got that all wrong. Anyway, you get the point. Mark of the beast. Mark of the beast, folks. And this is a barcode in your brain. If they can do it in your brain, they can do it in your wrist or your hand, which is what they're referring to for shot records. Quantum dot technology sewn into your skin that fluoresces under an ultraviolet or an infrared light so that you can safely travel around rather than getting a bracelet every day. You get your quantum dot vaccination record stitched into your wrist. But again, I'm digressing. I'm getting a little irritated if you haven't noticed that. This stuff bothers me to no end because we are at the end. We're very close, folks. We're very close. Anyway, let's go. Fluorescent in situ sequencing, FISQ. A highly multiplex technology for precisely locating multiple RNA molecules simultaneously within cells and tissues. Working copies of active genes called messenger RNA or mRNAs translate the genetic information present in DNA into proteins within cells, multiple compartments. I'm going to read that again. It's important. Working copies of active genes called messenger RNA or mRNA translate the genetic information present in DNA into proteins within the cell's multiple compartments. They are often positioned strategically within the cells in ways that contribute critically to how cells and tissues grow, develop, and function, and their mislocation can lead to disease development. So bad RNA can lead to disease. Misplaced RNA can lead to disease. To analyze many or all mRNA present in a cell or tissue samples simultaneously, scientists usually physically disrupt the cells, isolate the mRNAs, and then use next-gen sequencing methods to determine the exact sequence of the letters or base identifying each mRNA, the nucleotides A, C, T, and G. Genome sequencing provides a list of genes that are present. However, the valuable spatial information regarding where each mRNA is located within cells and tissue is lost. But wait, there's a solution. George Church, PhD at core faculty member at Wyss Institute and professor of genetics at Harvard Medical School, explains how fluorescent in situ sequencing could lead to new diagnostics that spot the earliest signs of disease and how it could help reveal how neurons in the brain connect and function.
Medicine is moving from very blunt instruments where you had to open up a chest all the way or had to use a molecule that would hit almost every part of your body to now molecules that are the finest scalpel you could ever have. They can find one base pair out of 6 billion and change it, for example. And so that needs to have matching it observational tools where we can see at that high level of resolution and comprehensiveness.
Well, just waiting for the 10th. I'm speechless at this point. Look at those eyes. Just look at them.
With current sequencing technology, you have to isolate RNA, and that usually means your first step is to grind up the sample, losing all spatial information of where the RNA molecules came from. With fluorescent in situ sequencing technology, we can actually detect the RNA transcripts where they live inside cells. And this is important because biological samples are not homogeneous. When we can detect where the RNA molecules are within the cells, that tells us about the function of those cells.
Please tune in next show, Friday morning, East Coast time. So we're going to start looking at CRISPR technology and look at RNA and DNA, and it's going to be fun.
For example, we could serial section through an entire embryo or brain. Specifically in the Brain Initiative, we want to encourage innovative neurotechnologies. This is an example of one that can, fluorescent in situ sequencing can integrate many different types of data at very high resolution. We now can break through the barriers to integrating the connectivity and the activity of the different neurons. This is a big
connectivity and the activity of neurons. Break through and understand. Thanks to taxpayer funding through the Brain Initiative, initiated by former President Barack Hussein Obama. And the program is still ongoing.
Big improvement over either the low resolution methods that we typically have and the super high resolution electron microscopy, which is hard to scale up beyond a few cubic millimeters.
With the new fluorescent in situ sequencing technology, we can visualize the sequence as a vertical string of colors with one color for each DNA base, A, G, C, and T.
Oh. So each color gets its own nucleotide, each base, A, C, T, and G. They can sequence it down to a single nucleotide in an RNA. One.
And then it appears that each molecule has its own sequence barcode. So
the molecule let's just for all intents and purposes call this a molecule, is the total sum of all of these fluorescent pixie sticks or rave sticks or whatever it is you want to infer these as. All of them make up one molecule, and they are imaging down to a single nucleotide with fluorescence, no less.
Its own sequence barcode. We can take those sequences and map them to the genome, and that tells us where each gene is expressed within the sample.
Fluorescent in situ sequencing could lead to a new era in the sense that we can really start looking at comprehensive expression and relationships among cells.
Look at that. You guys want to know what a barcode looks like in a brain? Here it is. Flashing lights. I like the analogy. What was it? Bright light toys. Beautiful. Exactly. Exactly. Barcoding your brain, folks.
Over vast spatial distances.
In this present study, we've detected over 6,000 genes in a single sample, whereas with current technology, we'd be limited to looking at 2 or 3 genes.
When you look at the whole set, you find the non-subtle parts, the place where the transcript you didn't think to look for is way out of proportion, right? So by looking comprehensively at all the transcripts, you have a better chance of finding the one that is causative or is a potential weak point where you can get a therapeutic in there.
Oh, a potential weak point where you can get a therapeutic in there. It's therapy. It's good for you. It's got to be good for you with a name like therapy, right? Maybe. We'll call vaccines therapy. We'll call GMO food therapy. We'll call fluoridated water therapy. Johns Hopkins, APL, and university students joined forces to map the brain. Researchers from Johns Hopkins University Applied Physics Laboratory partnered with Johns Hopkins University students this summer on a pilot program called CIRCUIT, Connectomics Institute for Reconstructing Cortex, Understanding Intelligence Together. 10-week summer internship program combined a sponsored research opportunity to map the brain to nanoscale resolution, intensive training, and mentor support. CIRCUIT, undergraduate students from a variety of academic backgrounds collaborated to help proofread brain maps that were generated through the Intelligence Advanced Research Projects Activity IARPA, Machine Learning Intelligence from Cortical Network program known as MICRONS. MICRONS is an applied research program by IARPA that seeks to reverse engineer algorithms of the brain and to revolutionize machine learning. All right. 15 minutes left. Let's continue.
With the help of students from Johns Hopkins University, the Johns Hopkins Applied Physics Lab is working on solving the mysteries of the brain. The CIRCUIT program, which stands for Connectomics Institute for Reconstructing Cortex, Understanding Intelligence Together, was an 8-week summer program that combined a sponsored research opportunity to map the brain at nanoscale resolution with intensive training and support.
So, more than one school, obviously, research institute, obviously, is working, no pun intended, on this program, right? So to put this in a broader scope, right, kind of going up a little bit in altitude, metaphorically speaking, when you look at the theft of intellectual property, it can be from many different institutions. So
in APL's Intelligence Systems Center on their main campus, students from underserved or underrepresented backgrounds in research will be helping APL scientists reverse engineer the brain.
So you see what she's doing right there? That's a HoloLens. And she's looking at augmented reality right now, walking around something that through the HoloLens is, looks like this, but it's in front of her, and she's pointing at it, right? They make these things. I just read a patent today, as a matter of fact. They have a VR headset that integrates 3 or 6 different sensors that read the brain at the same time. Using these bands, they can put sensors on these things and provide electroencephalographic information. And they don't have to be little metal electrodes you can see. They can be hidden beneath the plastic. Those of you that were with me when we went over the headphones, the Apple Series Beats by Dre headphones, those have electrodes on them that, well, go back and watch that one, folks. That'll blow your mind.
The brain.
What the students have been doing is building maps of the brain. So they're looking at synapses, they're looking at neurons, they're figuring out how to literally trace the pathways between individual synapses by hand and understand the connections that we all have inside of us.
I've never had an experience like this in my life. I'm studying neuroscience, but I didn't realize how much computer science is integrated into that.
It's critical. Computer science and neuroscience are they're in lockstep, hand in hand, going through this endeavor, this challenge.
The mentors were there every step of the way, but at the same time allowing us to kind of think about it and problem solve ourselves.
In enrolling in the program, they get training in neuroanatomy and get familiarity with some of the software tools that are used in the community and the data processing pipelines that are used in connectomics. And in reverse, they are producing really key data for our evaluation process.
APL's involvement with undergrads is especially important, and that's how I think the future will progress with science, is getting more and more people involved.
So it's mutually beneficial, right? They get to learn about the tools that are available, and then you get young, creative minds who have not been entrenched into the scientific bureaucracy at the higher levels to look at these problems, look at the current data sets, to then offer creative insights. It's a very useful thing. When you reach a stagnation point, because the teams that are working on a project have worked through the iterations over and over and over again, you hand off whatever their findings are to a completely novel group or a novice group. Give them a basic set of instructions and have them look at the task, and they will observe it and come up with conclusions and or paths forward that have not yet been realized by those that have been staring and working at the same problem for years in the past. So it's a win-win, so to speak.
The more brains you have around a table, the better answer you're probably going to get.
I came in as a pure biology and neuroscience major, hopefully going to medical school. This opportunity can allow me to mesh those two fields to help advance the fields of medicine, neurology, and other things in biology.
This pilot program was a great success this summer, and APL is already looking forward to continuing its legacy of sharing knowledge with the world for many more summers to come.
This is an applied physics laboratory. Johns Hopkins Applied Physics Laboratory is a national laboratory. And that's why you're seeing the augmented reality, virtual reality, whatever else they have at that school. They've got the latest and greatest. I'll put it that way. I don't want to speculate, but I wonder what sort of sensor arrays are in classrooms at the Johns Hopkins Applied Physics Laboratory. And anyway, let's move on. Another article about IARPA. I don't think we're going to read this. I want to continue. So it's in the show prep if you want to read about it. It's discussing finding the hidden algorithms of the brain, learning how the neural circuits work. Oh, your brain on a chip. How do you study something as complex as the human brain? Take it apart. WIS researchers have created organ chips. I'm all amped up, folks. Forgive me. That mimic the blood-brain barrier and the brain, and by linking them together, discovered how blood vessels and our neurons influence each other. So this is a little out of the scope of software, but this is how they get around human testing. They can model this stuff on chips.
Like all organs in the human body, the brain receives oxygen and nutrition from blood flowing through small vascular vessels. The blood vessels that supply the brain form a special structure called the blood-brain barrier, which is composed of endothelial cells that line the vessels, and astrocytes and periocytes on the outside that interface with neuronal cells in the brain. This barrier is highly selective and prevents toxins and other large molecules, such as antibodies, from entering the brain tissue, while allowing certain small substances, such as glucose, to pass through.
Nanotech. We're going to get there, folks. Nanotech passes right through this stuff. Notice they said large molecules, right? Off we go.
Nutrients and oxygen diffuse from the blood into the brain to feed the neurons and their supporting astrocytes.
So does nanotech.
Helping them function normally. Then hormones and metabolic products released from the brain cells diffuse back across the blood-brain barrier and into the blood, where they are transported away. To learn how the cells of the blood-brain barrier interact with brain cells, researchers at the Wyss Institute and Harvard Seas have developed a linked blood-brain barrier chip and brain chip system. The two blood-brain barrier chips each contain a vascular channel lined with human endothelial cells separated by a porous membrane from a parallel perivascular channel containing cultured brain astrocytes and pericytes.
You ever wonder where they got those from? I wonder where they got those from. They were cultured, right? Which means they probably had a starter cell and then they grew a culture of them and then took the culture, put it on the chip, or they grew the culture on this chip, or they grew it on a tray that then inserted into this chip. But where did they get the cells from to begin with?
The brain chip contains various types of neurons in one channel, separated from another perivascular channel in a semi-permeable membrane. The perivascular channel of the brain chip is microfluidically linked to the two blood-brain barrier chips, allowing scientists to monitor the flow of molecules between the brain and its blood vessels.
I'm sorry. Chat, you're hilarious. You got a couple there that are pretty good.
Just like in a living brain, small molecules such as glucose flow into the system through the vascular channel and pass into the perivascular channel, which leads them to the neurons in the brain chip. At the same time, small molecules released from neurons flow out through the same perivascular channel and into the second blood-brain barrier chip, where they pass through the endothelial cells and back into the vascular channel. Large molecules such as antibodies flowing through the vascular channel of the blood-brain barrier chip do not enter the perivascular channel or reach the neurons in the brain chip, again mimicking a living brain. Using this system, researchers discovered that the vascular and neuronal cells in chips that are fluidically linked produce proteins that are more similar to those naturally found in the human brain and its blood vessels than the proteins produced by cells in unlinked chips. In addition, by tracing the path of labeled glucose molecules, the researchers found that glucose metabolites produced by the endothelial cells cross the blood-brain barrier and enter brain neurons, where they are used to manufacture neurotransmitters such as glutamate and GABA. This intriguing finding suggests that the health of our brain's blood vessels could have a direct impact on the way our mind functions. Researchers are continuing to use this type of fluidically linked multi-organ chip system to uncover more about how the brain and other tissues and organs work at the molecular level.
So, it was a real nice watered down test. You really think they designed that whole thing like that just to look at sugar, folks? You really think, I don't. I think they would do things like, let's see what we can get to pass through the blood-brain barrier. What combinations? How do we encapsulate something to get it to think that it's safe to pass through? Only it's not, because there's something inside. What I'm talking about is an encapsulated substance. We'll get there. Before we close, these are church laboratory grant proposals. Harvard. This is his website, right? So grant proposals. See if it. Well, we're going to go back just to prove it to you, right? This is George Church, right? This is his bio. Clicked on Church. So now I'm going to go back because this wasn't, it's not in the top menu up here. I don't want to hunt around for it. Just going to look at this. Department of Energy Technology, National Institute of Health, Molecular and Genomic Imaging Center, 2003. He was getting grants all the way back from 1987. NIH, Causal Consequence of Variation, NIH, Genomically Engineered Organs. We'll click on that one. Genome Engineering, an iPSC model of Alzheimer's disease. NIH, Exploring a Novel Paradigm of Schizophrenia and Bipolar. Recording Neural Activities into DNA. Recording Neural Activities into DNA. We clicked that one too. Personal Genome Project, Cell Resources, Wyss Institute Synthetic Biology Platform, DARPA Biocompatibility, DARPA Living Foundries, DARPA Dialysis-like Therapeutic Systems, IARPA Microns Brain, which is what we looked at tonight, DARPA Safe Genes, DARPA Engineered Living Materials, and IARPA Functional, Genomic, and Computational Assessments of Threats. So we have a few minutes. We're going to browse these real fast. Genomically engineered organs. The organoids, to make organoids more like real organs, they must be vascularized. Vascular channels can be engineered into tissues by 3D printing techniques that lay down channels so they can be lined with human endothelial cells within a gel matrix containing human fibroblasts. Cell printing is shown on top in a print head, laying down one of 3 kinds of cells in a pattern. Now, if you explore the Wyss Institute's website, you will find this in action, videos of it. We want vessels to grow in the organs and the organoids, and so are developing ways of leveraging natural propensity of endothelial cells to sprout new vessels, shown at the bottom. So they can 3D print and have these things self-replicate and then grow them into tissue and create vascular structures inside of engineered tissue for artificial organ replacements. Oh, they have all these other things too. Expansion microscopy, we looked at that today. We looked at barcoding your brain today. And super high resolution Ogliostorm, which looks not so fun. Let's move on. We have 1 minute left. Recording neural activities onto DNA. Progress in neural recording is critical to understanding the brain. Current neural recordings can be at best capture a few hundred interacting neurons. This is in the show prep if you want to go exploring his website. George Church, PhD, Epstein affiliate. Safeguarding genes. 3 teams at Harvard Medical School are launching a project to support responsible innovation in gene editing and to protect against threats to genome integrity. You can read about this if you wish. We are out of time, folks. We are out of time.